Okay. So hello everyone. My name is Naibedya Chattopadhyay. I'm not Nivedita. Well, before I begin this presentation, let me profusely thank ISBMR for giving me this opportunity to deliver the oration in memory of Professor Teotia, an outstanding clinical endocrinologist of our country had. His original research on metabolic bone diseases, including hyperparathyroidism and osteoporosis, is extremely valuable. His contribution to bone histomorphometry, kidney stones, and fluorosis have indeed immensely enriched the field of metabolic bone disease. In particular, that stupendous effort of epidemiological having the map of epidemiological map for fluorosis and bone disease was breathtaking. So these contributions are simply humbling, and I will try to give some justification for inviting me to celebrate Professor Teotia's research contribution through this oration. Now, I can't see the hall, but I assume that ISBMR is particularly dominated by clinicians. So I'm going to be speaking to you today about something you are always looking for to treat your patients who are suffering badly from osteoporosis. Her T-score is ranging from, suppose, minus 2.5 to minus 3.5 and suffered fractures, but she cannot afford teriparatide. And even if you buy it for her, she cannot use it because she does not have a refrigerator, as she belongs to one of 70% of Indian households that do not have it. So our research may give you a potential alternative to teriparatide. So this is a mandatory conflict disclosure that has no bearing on today's talk. For the next 40 or 45 minutes, you will know these things: working knowledge on phosphodiesterase, what are phosphodiesterase inhibitors, why are they relevant to bones, and I will give a few examples from my lab on the skeletal effects of phosphodiesterase inhibitors. Now, I will talk only on those phosphodiesterases that cleave phosphodiesterase bonds of cyclic nucleotide. I will not cover other members of phosphodiesterase superfamily such as DNase, RNase, sphingomyelin phosphodiesterases or phospholipase C and D, and autotaxin. Now, this diagram shows that from ATP and GTP, respectively, cyclic AMP and cyclic GMP are formed by the action of adenylate cyclase and guanylyl cyclase, respectively, and then they then in turn activate pKa and pKg, and this in turn will activate transcription factors leading to gene expression. And these phosphodiesterases will desensitize or degrade the cyclic AMP to inactive AMP and GMP. Now, these phosphodiesterases are metallohydrolases that control the concentration of second messenger cyclic AMP and cyclic GMP. And basically, these AMP and GMP are 5' inactive metabolites that are formed due to the action of this phosphodiesterase, which are 11 total in number, which is in contrast to hundreds of kinases. There are hundreds of kinases, there are only 11 phosphodiesterases. Now, remember that a short burst of cyclic AMP or cyclic GMP in osteoblast is bone anabolic, and it is a very important feature of phosphodiesterase inhibitors to act as bone anabolic. Now, this is a schematic diagram showing the mechanism of cyclic AMP-induced osteogenic events. So, for example, signaling through PTH or BMP will increase will increase cyclic AMP by a cascading mechanism, and that would lead to activation of cyclic AMP responsive element binding protein. On phosphorylation, it will enter into the nucleus and will transcribe the osteogenic genes that would lead to osteoblast differentiation. Now, where is the link between phosphodiesterase and PTH-R1 signaling? And that link lies in endosome. So PTH will act through type 1 PTH receptor on the membrane, this is G-alpha coupled GPCR, and that will stimulate adenylate cyclase cyclic AMP production. But once it gets internalized by endocytosis, that is the way, as you know, that these GPCRs most of the time get desensitized and they're internalized. And while inside the endosome, these complex receptor ligand complex continue to signal, and the signal is actually at that time is activation of ERK1 to a MAP kinase, and that will inhibit PDE4, one of the major PD isozymes present in osteoblast. In this regard, I will tell you that among the 11 phosphodiesterases, PD4 and PD5 are the most abundant isozymes present in osteoblast. So by inhibiting PD4 by ERK1/2, the cyclic AMP is prevented from getting degraded immediately. And that is where the PD signaling and PTH-R1 signaling merges. I will show data to bring this point home. Now, this is the slide showing the mechanism of cyclic GMP-induced osteogenic event. In osteoblast, mechanical shear-generated fluid shear stress is one of the major cyclic GMP activator. Besides insulin, estrogen membrane receptor, not the nuclear, membrane receptor, and C natriuretic factors, which will stimulate NOS, nitric oxide synthase, and ultimately cyclic GMP will be produced, and that activated cyclic GMP will then stimulate protein kinase G, and it will stimulate a separate bunch of osteogenic genes than the PKC TREB activation. This would be COX-2, EBP beta, AP1, FOS, and Jun. And the effect would be osteoblast proliferation and survival. So having provided the background of PDE inhibition and osteoblast function, I will now begin sharing our data with you. We screened 21 clinically used phosphodiesterase inhibitors in a library, which we have, which is an FDA-approved drug library containing 1,300 plus drugs. The screening system is phenotypic, in which we measure ALP as a surrogate of osteoblast differentiation from the primary rat osteoblasts or mouse osteoblasts. 16, these are the 16 phosphodiesterases had no effect on osteoblast differentiation. 2 inhibited it, these are theophylline and aminophylline, and 3, pentoxyphylline, sildenafil, and vardenafil, increased it. So I will discuss briefly about theophylline. Now, if you see, both the drugs caused loss of osteoblast viability, with IC50 of theophylline being 98 micromole and aminophylline is almost 5 times more than theophylline. So we studied theophylline in some details. Now, what is important here to note the kinetics and the profile of cyclic AMP stimulation by theophylline. It has been compared with PTH, forskolin, which is an adenylate cyclase activator, and the vehicle. And when the time course was done, you see there is a sharp increase at 30 minutes of cyclic AMP by theophylline, and that continues to remain high, and compare it with PTH. So when we did the AUC, you see that it is several fold, even at least 2 folds higher than PTH and phosphodine. Now, when you have this sustained increase in cyclic AMP in any cell, including, of course, osteoblast, the cells undergo apoptosis. And so the viability of osteoblast was reduced by theophylline, and a pKa inhibitor, KT drug, KT compound, that actually reversed this effect, which suggested that it is pKa dependent. And then theophylline induced ROS production, and glutathione suppressed it, and so also did KT. So this suggested that it is a pKa dependent ROS, partially ROS mediated, but fully pKa dependent apoptosis inducer for osteoblast. Now, in this regard, I would like to give a little history. As you know, that it is said that necessity is the mother of invention. And in this case, Henry Hyde Salters was the inventor of PDE inhibitor. The first batch of PDE inhibitor was isolated by him, and incidentally, he used to suffer from asthma, and he died at an young age of 48 years. And he was very fond of coffee, particularly because it relieved him from asthmatic attacks. And the xanthine compounds, caffeine is also xanthine, and theophylline is xanthine, were discovered as the first generation cyclic nucleotide PDE inhibitors. It was done in the mid 19th century. And this paved the path for discovery of one of the largest sources of clinically used drugs for a variety of diseases other than asthma, from which Henry Salters was suffering. Now here comes the data on the issue of, and after showing this, what I would like to show is the effect of theophylline in vivo, in rat. Here is the data for in vivo. Now, when the rat dose was human dose was converted to rat dose, it turned out to be 100 milligram per kg. And normal adult rats were given theophylline at 100 milligram and half the dose 50 milligram for 2 months. And we also kept methylprednisone, because generally the asthma patients are also given methylprednisone very commonly. And methylprednisone is a well-known drug causing osteopenia. So what you see here is that at 2 months, methylprednisone 3 milligram per kg significantly reduced BMD and bone volume. BMD, because this is volumetric BMD, because this is data from micro-CT. And volumetric BMD was significantly decreased, and theophylline at 100 milligram dose inhibited both BMD and bone volume at female, sorry, it would be metaphysis. I wrongly written epiphysis. And then the mechanisms were studied. And what we found is that at 2 months, theophylline resulted in significant, almost twofold decrease in serum PINP, the osteogenic serum marker, and increased threefold increase in CTX1, the resorption marker. And when we did the bone histomorphometry, this dynamic histomorphometry to measure mineralizing surface, mineral apposition rate, and bone formation rate, all were significantly decreased by theophylline. And besides, there is a tremendous increase in resorption, which the CTX1 is reflecting. And in the bone, the OPG rankle ratio was tremendously down, just because the rankle was so much stimulated. And rankle gets stimulated because cyclic AMP is stimulated, because rankle promoter contains Krebs binding element. As you know, PTH also increases rank ligand, and that is the reason why there is a loss of anabolic window by PTH. It's a very similar thing is happening with the opposite. At this case, actually, at this case, it is a similar thing, but when it comes to the bone formation, it is opposite of PTH, and that is because of these tremendously elevated cyclic AMP. Another insight caused by this theophylline, that it reduces hepatic vitamin D synthesizing enzyme. Serum vitamin D is significantly reduced in the animals giving theophylline, and the hepatic vitamin D producing enzymes such as CYP27A1 and CYP2R1 at even 4 weeks. See what a huge downregulation even at 4 weeks has happened in these animals given theophylline. But the good thing is that if these animals are given vitamin D supplementation, the theophylline treated animals, then there is a significant reversal or protection from osteopenia. So the bottom line is that that if any asthma patients are given theophylline or had to be given theophylline, it has to be ensured that vitamin D is prescribed, even though she is she has normal level of vitamin D, because it is expected that with time she might become hypovitaminosis D condition. So the conclusion from this theophylline study is that chronic theophylline treatment induces severe osteopenia. That is because of inhibition of osteoblast function, and that is because primarily osteoblast apoptosis and concurrent stimulation of osteoclast function. There is a decrease or suppression of hepatic synthesis of 25 hydroxylases, and supplementation of vitamin D significantly protects against theophylline-induced bone loss. And this study we published in 2016. Now, the second group of drugs, pentoxifylline, vardenafil, and sildenafil, that increased ALP. So pentoxifylline is a pan-phosphodiesterase inhibitor, and it is a methyl xanthine structure. And what it does is that it has a increase. It causes increase in ALP at 10 and 100 nanomole. And when we did the EC50 calculation, it comes to around 8 nanomolar EC50. Now, take a look at this profile of cyclic AMP profile. So PTX, pentoxifylline, and PTH, when the osteoblasts were treated with these two drugs, they basically superimposed, which is in stark contrast to what we saw with theophylline, that superstimulated cyclic AMP levels. And this is the AUC. And so that is why I kept on saying is that the kinetics and the extent of cyclic AMP induction is the key. Now, it stimulates osteogenic effect. That means in vitro nodule formation by bone marrow stromal cells, where by PTX were stimulated, and the osteogenic genes RANX2 and COL1 were significantly upregulated by pentoxifylline. Now, it promotes osteoblast differentiation via cyclic AMP PKA pathway because when the cyclic AMP and PKA inhibitors SQ and H89 were used, the pentoxifylline induced osteoblast differentiation assessed by ALP were all abolished. And then the CREB, the transcription factor, which is upstream of various osteogenic genes, where this pentoxifylline caused a rapid phosphorylation of CREB, starting from around 30 minutes and lasting up to 90 minutes. And then H89, the PK inhibitor, abolished this effect, which suggested that the pathway is cyclic AMP PK CREB RUNX2 pathway, particularly when we saw that by 48 and 72 hours, RUNX2 protein went up threefold at least by pentoxifylline, and presence of H89 PK inhibitor blocked this increase. Now we studied the effect of PTX in adult animals, and when we converted the human to rat dose, it came to 150 milligram per kg. And then we used one third of that and 1/6 of that in animals and assessed their PK. And what we found is that the Cmax comes to around 7 nanomolar. And recall the EC50, in vitro EC50, osteogenic EC50 of pentoxifylline was 8 nanomolar. So the PK data suggested that we could use even 1/6 of the adult human dose, which is 25 milligram per kg in animals. So we did this biomarker profile of PTX and PTH. We did the comparison in overectomized osteopenic animals. So the animals were first made osteopenic. And then we did a time course of PINP and CTX. And what you see here is that there is no significant difference between OVX and sham animals, that means the ovary intact and overectomized animals, up to 4 months. But PTH, as usual, as expected, significantly increased it, and so was PTX. And in case of CTX1, the resorption marker, both PTH and PTX kept it elevated like OVX animals. So which suggests that like PTH, pentoxifylline is a pure bone anabolic agent. And when we studied their trabecular bones, both PTX and PTH completely recovered the BMD and BVTV at femur metaphysis at L5 vertebrae, fifth vertebrae. And then we did a L5 compression test, and we found that the maximum power, energy, and stiffness, the 3 indices by which we measure bone strength, were all significantly decreased in the osteopenic animals, whereas PTX recovered it to the control level. Now, this is the trabecular dynamic histomorphometry, which is the gold standard for anabolic effect. And what we found, this is from femur metaphysis. And what we found is the bone formation indices of pentoxifylline were equivalent to PTH. Mineral surface, mineral apposition rate, bone formation rate, all were similar. And most notably, PTX and PTH had significantly higher bone formation rate over the ovary intact animals, which actually suggests that it is it is on a par with PTH. The anabolic response is on a par with PTH. And then we studied the bone quality. This is actually bone crystallinity is measured by XRD, X-ray diffraction based full width half maximal. In simple words, higher FWHM reflects lower crystallinity and thus indicates poor crystal packing. Now, FWHM values in the femurs of OVX plus vehicle were significantly higher than the sham, and PTX and PTH diminished it and brought it to the level of sham. Next, we measured calcium weight, tissue calcium percentage in the sections of femur metaphysis by backscattered electron microscopy and found that these osteopenic animals had significantly lesser calcium weight than the sham group. And strikingly, both PTX and PTH increased it more than the sham. And that actually explains that how these two drugs confer greater stiffness to the bone. Now, one concern might come that if it is causing greater stiffness, is it also inhibiting ductility or increasing brittleness? Unfortunately, we have not studied that. Then we studied the angiogenic effect of this drug, because angiogenesis is a critical event in bone formation, particularly at the trabecular sites. And VEGF, vascular endothelial growth factor, importantly contributes to this process. Loss of capillary bed and age-related decline in skeletal vascularity has been linked to osteoporosis in humans. And because pentoxifylline induces intercellular cyclic AMP that has a supportive role in vascular physiology, we surmise that along with cyclic AMP-dependent stimulation of osteoblast function, pentoxifylline could also act to improve skeletal vasculature. So this is a fluorescent signal indicative of bone vascular volume of femur and tibia, and that showed significant reduction in the osteopenic animals, and it was significantly, it was brought to the level of the sham by pentoxifylline. And then in the bone, VEGF levels were significantly decreased. This is the Western blot analysis, the VEGF levels and its cognate receptor, VEGF-R2, both ligand and, but although it looks here like some difference, but actually it is the VEGF that was inhibited, did not reach significance. But what happens that when we give PTX, that actually stimulates to a great extent, almost doubles the expression of the ligand as well as the receptor, which might explain why by pentoxifylline there is a significant increase in the vascular bed in the bone. So the conclusion of the pentoxifylline study is that this drug completely restores trabecular microarchitecture and strength in osteopenic rats and rabbits. I have not shown you the rabbit data. At the 1/6 human equivalent dose, pentoxifylline acts by increasing osteoblast function, but does not have any effect on osteoclast. It has proangiogenic effect in bones of osteopenic animals, and bone crystallinity and calcium percentage is improved by PTX, suggesting improvement in the bone quality. So what is the future direction? A comparative study between pentoxifylline and teriparatide for the treatment of postmenopausal osteoporosis, a randomized controlled trial. And this proposal was funded by ICMR to Dr. Babu Garg at AIIMS New Delhi, and it was actually funded last year in May, but due to the COVID situation, the money has not yet been released. And my most recent conversation with Dr. Garg is that they're on the process of finally releasing it. And then the study, to study the osteogenic potential of pentoxifylline metabolites, there are 3 metabolites, and 2 of them, lysophylline and a propyl hydroxy derivative, both have PD inhibitory activity, and that needs to be assessed. We published this data, one in Calcified Tissue International, and the rabbit data was published in Bone. Now the last segment, in vitro osteogenic screen of FDA approved drug by we have discussed pentoxifylline, but now that was a pan-PDE inhibitor. Now we are moving towards specific inhibitors. That means PDE5 inhibitors, because PDE5 is one of the most abundant PDE isozyme present in osteoblast, and the drugs we tested are vardenafil and sildenafil. Now, sildenafil, actually it is a pyrazolopyrimidinone, and vardenafil is a benzosulfonamide, and both drugs stimulated ALP activity to almost equal EC50 level of around 100 nanomolar. And then we checked their in vitro osteogenic nodule formation effect using bone marrow stromal cells, and both at their EC50 values significantly enhanced nodule formation, increased osteogenic genes, RUNX2 and COL1. And then sildenafil and vardenafil being a CGMP coupled signaling molecules, it did not increase cyclic AMP, PTH increased in this assay, but cyclic GMP was strongly upregulated by sildenafil and vardenafil. PTH had no effect in cultured osteoblast. And then we used NOS inhibitor, LNM, and the cyclic AMP inhibitor SQ. Only the NOS inhibitor LNM blocked the drug-induced, sildenafil or vardenafil-induced osteoblast differentiation. Both VEGF-R2 and VEGF, in this case, both of them were significantly upregulated by sildenafil and vardenafil at 72 hours. And LNM, the NOS inhibitors, abolished the effect on VEGF by both sildenafil and vardenafil, which suggests that the pathway being cyclic GMP dependent, NOS dependent VEGF signaling being upregulated by sildenafil and vardenafil. So this is the slide to show that there is a coupling of osteogenesis and angiogenesis by VEGF. That whenever there is a low blood supply due to the loss of vasculature to bone, there would be low oxygen tension, and the low oxygen tension will lead to increase in the HIF, the transcription factor, hypoxia inducible factor. That will then stimulate VEGF, and VEGF will lead to increased angiogenesis, vasculature formation, as well as osteogenesis. So we studied the effect of sildenafil and vardenafil in osteopenic mice, and in femur metaphysis, you can see that there is a remarkable loss of trabecular bone in the osteopenic animals, and sildenafil and vardenafil restored it to the level of PTH. And when we studied their vasculature by in vivo imaging system, we found that osteopenic mice had significant decrease in vascular bed, and sildenafil actually is the only one stimulated it even more than the sham animal. Now, vardenafil had brought it back to the sham level, but it may be of interest to you that PTH also increases vascular bed compared to osteopenic animals. In the bone, what we found is that the Western blot analysis of the bone revealed that VEGF and VEGF-R2 were significantly decreased in osteopenic animals, and sildenafil and vardenafil, as well as PTH, brought it back to the sham level. And sildenafil is the one which was even more than the sham, the VEGF, the ligand. So it seems that the vascular effect of sildenafil is better than vardenafil or PTH. Now, when we used sunitinib, a blocker of tyrosine kinase that also blocks VEGF-R2, the bone restorative effect of sildenafil and vardenafil were completely lost. As you see that the BMD and BVTV were blocked, the stimulatory effect was blocked by sunitinib. So the conclusion from this last part of the study is that both drugs, sildenafil and vardenafil, promoted osteoblast differentiation, but sildenafil was more potent in stimulating mineralized matrix formation. Both stimulated osteoblast differentiation via NOS cyclic GMP PKG pathway, increased both ligand and the receptor expression in osteoblast. Both completely restored trabecular microarchitecture by pure osteoanabolic mechanism. I haven't shown you the data of osteoclast because it was a negative data that both drugs had no effect on osteoclast function. Although cyclic GMP has shown to have some role, but it is something which is not yet been confirmed. I mean, the data are equivocal, and there is increased angiogenesis and VEGF expression in osteopenic animals. So this drug induces bone formation or the anabolic effect by both osteogenic as well as angiogenic effect. This study was published last year in Bone. Now, note one thing, that this study came online on 29th February 2022. And then in June 2020, this study was published in PNAS, repurposing erectile dysfunction drugs, tadalafil and vardenafil to increase bone mass. It was contributed by Maria New, who is also a co-author to PNAS, on April 17, 2020. And our paper came online almost 1.5 months before. And of course, our paper was not cited. But what you should note here is that they found that both agents, tadalafil and vardenafil, resulted in bone formation. And at the same time, it blocked resorption, which we did not see. We only saw a pure anabolic effect of sildenafil and vardenafil. So the summary, overall summary, is that theophylline has osteopenic effect. Pentoxifylline has a non-selective, which is a non-selective PD inhibitor, is osteoanabolic and osteoangiogenic. And so are the PD5 inhibitors, both osteoanabolic and osteoangiogenic. So the future direction would be to have new drug series based on PD4 and PD5 co-crystallized with pentoxifylline, which is a pan-phosphodiesterase inhibitor, or rolipram one may take, which is a PD4 inhibitors, and a previous paper has shown that rolipram has bone anabolic effect, and PD5 inhibitors, sildenafil and vardenafil. We have actually undertaken this preparing new drug series based on the co-crystal structure of PD4 and 5. The work is in progress. Novel formulation with bisphosphonate to suppress, to actually what I meant here, it is not properly sentenced. What I meant is that one could think of giving a combination of bisphosphonate and pentoxifylline to stimulate bone formation and at the same time inhibit bone resorption. And then clinical trial of pentoxifylline in both postmenopausal osteoporosis and glucocorticoid-induced osteoporosis. Postmenopausal osteoporosis is in line, is going to start soon. But GIOP is I don't know if anybody would be interested in doing that. And checking the BMD of erectile dysfunction patients who are taking PDE5 inhibitors. That would be interesting to see if those taking PDE5 inhibitors, the erectile dysfunction patients, whether they their bones benefit from taking these drugs. Actually, the future direction we have discussed very broadly and elaboratively in a review that published this year in European Journal of Pharmacology. So if you want to know more about the future direction which we have suggested, you can read this review. These are the 5 persons who in my lab who were involved intimately with this project. Shubhashish, his PhD thesis had a significant chunk of the presentation I have given you today. And Konika is working on the new PDE4/5 inhibitors, the synthetic series that we have synthesized. And Wahajuddin was the one, my colleague actually, he is not from my lab, he is from pharmacokinetics department. He is my colleague, scientist colleague who has done the PK studies. And Sham and Monica has done the histomorphometry studies. And we have been generously funded by Department of Science and Technology and CSIR. Thank you.
Thank you, Dr. Chattopadhyay. That was fantastic. I mean,
thank you.
Dr. Teotia would be very, very happy with the work that you've done. And as a part of the deciding factors in choosing you for the oration, I think we've done a great job. Thank you very much once again and wish you all the luck and further accolades to you. Thank you.
Thank you.
Thank you, sir. As this is an oration, we don't have any questions, but there's a plaque which will be sent over to Dr. Chattopadhyay at his place in Lucknow, and